Okay, so help me out. From yesterday, was everybody here yesterday?
I was. I at work.
Okay.
I think everybody was.
So a couple things that Mr. Wagner had told myself is the materials that are on the overhead, are they not matching up on your paper? Yes, they are, or yes, that is correct.
That's correct. They are not.
Yesterday,
Okay.
I tried watching the thing and half the notes weren't. I couldn't.
Okay.
The notes weren't on.
Okay. Well, I tell you what we're going to do then. This will be. It's way too important. This is one of the things that and that's what I had mentioned to parents last week. If it was at this time 1 week ago, or 2 hours, 3 hours, 4 hours later than this, when it came to parent-teacher conferences, it's just way too important of a concept to say that would be an easy thing to do. Just say we're going to eliminate this from holding you accountable for understanding. Okay, so then one of the things I sometimes think, and it's just general response that you're going to want to say, let's just not do it. Okay, so yeah, that would be the easy way to do that. And yeah, I know you were. Like I said, I think that's just a general response. And I could say the same thing if that's something that you don't like doing. I could say I guess you don't have a problem with me. You have a problem with the state of South Dakota. And I think that would be accurate then as well. Okay, so one of the things, if you don't have a book, did that activity at least make sense?
Yes.
Okay. So still, go ahead and grab a book if you have not done so. Okay, 337. Now, this is going to seem odd moving forward in this curriculum that the chapter numbers will go down after this. Okay? So I think this is chapter 12. We'll actually go backwards. And the reason for this is I disagree with how the author presents this, because this is maybe something that you had covered in middle school as well. So I'm just going to put some general pictures up here. And I wouldn't draw this. Typically we say you might want to write this down, but that would not be the case here. Okay, in other words, these black squiggly lines are this. Ever heard of chromosomes before? Okay, so then my math would say 46, or 23 pairs, however you want to look at it. Oops. I guess I meant to do that, not circles. And like I said, I wouldn't write this down. We're just trying to make sense of why it is we make and take the order that we do. Okay, and then I ran out of space. From your limited knowledge of chromosomes, Can you name what this might be? And the process is called mitosis. Have you ever heard of that before? Has something to do with the cell. Yeah. Say that again.
Cell replication.
Yeah, that's, you're not too far off. Okay, hold that thought. Okay, so when you say cell division, did you say replication?
Replication.
Okay, so yeah, it's similar where this maybe even looks familiar. Do these terms look familiar?
Yes, they are.
Okay, those are all different phases of mitosis. So the reason that we do this, okay, is because when the cell goes through all of these phases, I'm just picking maybe chapter 8, chapter 9 is perhaps when this takes place, okay? This is chapter 12, and the reason that we're doing this now is because in them earlier chapters they talk about DNA replication. Well, why would you talk about that DNA replication in chapter 8 if you haven't even covered it yet? Does that make sense? So that's why these chapters are going to be what you might think as being out of order. Catching up on our Netflix or something, or what?
I don't think they allow Netflix on the computers.
They do.
They do?
I thought it was locked. It's blocked or something.
Netflix is blocked now?
So should I be under the presumption that what you're doing is way more important than this? And maybe it is.
I never implied that. I'm sorry if I did.
Okay.
I'm also sorry, but I'm just, it's in the background.
Oh, okay. And maybe you could just say your computer screen was open and I would believe you. You don't have to say, oh my gosh. And then when you say it that way, it's saying, now you're telling me that did catch you. Okay. All right. So the reason, again, that we talk about this is DNA has to replicate and go through that process before you can discuss all of this. So it was our determination that's not going to make any sense if you don't know what DNA replication is. Okay, so in this case, we want to, on page 337, is look at what the author talks about what transcription and translation is. Hopefully, we can give you a good diagram and or explanation on what this process is, and we'll try to show you why it's such an important process. Okay, page 337. Okay, the first step of the central dogma, I never understood why they say that. Dogma just means it's just a process or
37 or 77?
337.
did I say 377?
Oh,
Okay.
I'm smoking something.
Okay. Okay.
You cannot say that. You cannot say that. You can't say that, Tom. I'm on, no.
Okay. Once we come back to Earth here, okay, involves the synthesis of messenger RNA. That's what the M stands for, messenger RNA, from DNA in a process called transcription, okay? And the way I like to describe that is, you're going to say, gosh, he's all over the place here. Going over this, but that's in the future, to try to make sense of this. If you were to look at the English feudal system, you would have serfs at the very bottom. Then you'd maybe have priests above them. And in every parcel of land, I think in this feudal system, wasn't the knight all the way at the top maybe? And is this world history? Does this make sense at all?
Yeah. A little bit.
You're not just saying that. Who said yes?
Me.
Okay. So then maybe you would have clergy here or priests. I put clergy. I might not even spell that right, but I don't know if I can spell priests right, and I didn't want to do that, so I put this down here. So who would these people be? Yeah, exactly. These are at we'd say at the bottom here, these are not slave workers, but just the idea, the ones that did all of the manual labor, okay? So the importance of clergy members was then not that that was part of the religious faction, but they could do something with language that these people could not probably. Not speak, you're close.
Having the re-education.
Someone said read, that's correct. But more importantly, besides reading, they could write, okay? So, a different way you could look at these clergy members is maybe we call them scribes. A scribe would be someone who can do what? Not just write, but more importantly, they could read, okay? So if a scribe can read, we make the correlation of that's what transcription is. It's a process of reading the DNA, okay? So as we go farther, okay, the messenger RNA from DNA is a process called transcription, okay? Through transcription, the DNA code is transformed to messenger RNA in the nucleus. The messenger RNA can take the code into the cytoplasm for protein synthesis. Follow along with the process of transcription. The DNA is unzipped, just like that of your double helix molecule. Did you go through, and it's sad, I gotta mention this, not on your part that it's sad. It's sad I gotta go through this because it's been over a week since I've had in-class time with you. Because of athletic events, it's gotten in the way. Parent-teacher conferences has gotten in the way. But do we have a good understanding what DNA replication is? It would have looked something like that.
Yeah.
Okay. So this process is somewhat similar to DNA replication. Okay. The messenger RNA can take the code into the cytoplasm for protein synthesis. Yes, we read that. Follow along with the process of transcription. The DNA is unzipped in the nucleus in RNA polymerase. Now, once you hear polymerase, we should correlate that what type of substance is that polymerase?
It's an enzyme.
It's an enzyme because a lot of enzymes that we talk about in the human body will end in ASE, whether it's helicase, polymerase, amylase, lipase, all those are the different types of enzymes that you would see within the human body. Okay? The enzyme that regulates RNA synthesis binds to a specific section where messenger RNA can be synthesized. As the DNA strand unwinds, the RNA polymerase initiates messenger RNA synthesis, okay, and moves along one of the DNA strands. Don't worry about the 3' and 5' direction. That's more so of a way higher correlation of application than what we want to do at this level, okay? The strand of DNA that is read by the RNA polymerase is called the template strand, and messenger RNA is synthesized as a component to the DNA nucleotides. The DNA strand not used for the template strand is called the non-template strand. The messenger RNA transcript is manufactured into the 5' and 3' direction. Again, don't be concerned about that. Adding each new RNA nucleotide to, okay. Uracil is incorporated instead of thymine as the messenger RNA molecule is made. And I noticed that there's a couple diagrams up on the board where you were given that instruction that uracil replaces that thymine, okay? Then the messenger RNA is released to the RNA polymerase detaches from the DNA. The new mRNA then moves out of the nucleus through pores into the cytoplasm. Now, the reason that is important is because way back from chapter 7, okay, does this term look familiar? Something that's going to come full circle. Okay? Something very specific happens at the ribosome. Proteins are made, okay? That's where the proteins are assembled and made, is at the ribosome. And because we want to state it that way, is the ribosome in the nucleus? No, because we just read, what does the messenger RNA do? It leaves what structure? It leaves the nucleus. Where do you suppose it's going to go? It's going to go to the ribosome. Okay? So,
How does it know how to do this?
What do you do to breathe?
probably what I want to call the receptor sites that would be located on Did you see that 3-letter code? Okay? Now, if you're really good, you could draw the amino acid structure. Oh, no, wait, that's biochemistry. That's senior level, unless you want to learn that. You can, this will come full circle again. For those of you who do take biochemistry, rather than just looking at these as 3-letter base pairs, you will actually draw the structure of these amino acids. Because, again, I don't know how much we're going to get to this. It's a facial surgeries that we look at in how a lot of this protein synthesis, when it goes wrong, it can go wrong 5 different ways from, we hate to say it, 5 different ways from Sunday, but that's just a common phrase that happens because typically in lower developed countries that can't fix these problems, these different types of, if you want to call it traits or processes, they just don't get fixed because who do we have out here for farmers? I don't think there's anybody out there, is there? But what are you doing?
We're just looking at it.
Okay. So then, do you suppose it's a good idea to sit there? Okay, well, all right. Okay, so typically what happens, and we'll talk about this in the genetics chapter, is that a farmer that has a whole set of stock cows, eventually, why would they want to replace the bull? Because after about maybe 2 years, you're going to want to introduce new genes, okay? Because that genetic recombination is healthy for a herd. Well, what happens in some of these third world countries that these mutations that actually take place, they can't be fixed and they continue to show up in their gene population, okay? That's not the case that what we see here in a developed country like here in the U.S. Okay, so now I want to skip down on page 338 to translation, okay? Because these processes, okay, are related, but they're not exactly the same. Okay, so translation on 338. Once the messenger RNA is synthesized and processed, it moves to the ribosome. It's no coinkydink, we wrote that up on the board. In eukaryotes, which means cells that have a nucleus, okay, this means the messenger RNA must leave the nucleus and enter the cytoplasm. Once in the cytoplasm, all that, we don't need to know those 5 and 3 numbers, the messenger RNA connects to the ribosome. That is where the code is read and translated to make a protein through a process called translation, okay? So, one way to look at that is, okay, remember, what does a scribe do? Reads. So if the scribe does the reading, what does it mean to translate something?
Turn it into a different language.
Yeah, exactly. You're going to change it to a different language. So what we're going to illustrate here, and I think this is the best way to look at things, okay? And that is we're going to put everything together here. Okay? So in other words, we're going to make this a little bigger. Here's our ribosome, which, that's also a term, ribose. Okay? What's happening at that ribosome? Protein synthesis. Now, we explain this the same way every year because it works. Once you find something that works, okay? If you have a particular running play, or if you have 5 serves in a row, and they're all, not that they're aces, but they don't return them, wouldn't you not hit to the same spot? You'd be silly not to do that, right? Okay? So if you find a good running play and it's working, if you can run right all night, are you going to start running to the left?
Why not?
No, Tom,
Oh.
you're not. That is
What if they start blocking that up?
Well, but if we just said if you can run to the right all night, why would you go away from that?
Well, there's a kid like down
Okay.
on the right side.
Okay. So, this here is our nucleus. We're just making it bigger. Okay? So then, in a correlation, we call this, okay? And then our nucleus is also this. Okay. So when it comes to calling plays in football, who does all the running from the sideline to the huddle?
Squirtle.
Jackson and Wyatt.
Nope, they didn't want it.
Sometimes Jackson.
Just, who is it?
Wyatt. It's
Okay,
Wyatt.
Wyatt. I didn't think that was that difficult. Okay, so the sideline. Where does Wyatt get his instruction from?
Let's go. Offensive coordinator.
Coach Johnson.
Are we okay? I mean, we had a conference last week to make sure, do we need to have more communication to say, I don't know if you're okay? Okay? So, I need a coach. Okay, so I'm just going to put CJ for Coach Johnson. Okay? He's got the message. Okay? So, who is he going to tell that message to?
Wyatt.
Okay? So then Wyatt will leave the sideline because he has the message, correct?
Yeah.
So once he gets out here to the ribosome or the huddle, he's going to say what the play is. And then what does that play tell each and every individual to do on offense? It tells them exactly where they need to go. Okay, so on the sideline, here is our instruction. It would be, give me some nucleotides.
And, what's
Serene.
Serine is probably a amino acid. Thymine, adenine, cytosine.
Arginine.
You're giving me amino acids. We don't want amino acids. Okay, 3 more.
Guanine.
Guanine.
The U one. U.
No. So how about adenine and another cytosine, GAC or GAG or whatever. Okay, so our corresponding pairs then, adenine, thymine, guanine, okay, cytosine, thymine, and then guanine again. So we know what type of strand is this. For 2 reasons. We know it is 3 reasons. We know that this is DNA. What might be one reason we know this is DNA? Because it's got thymine on it. Another reason.
They have pairs. They're matching.
No. Where's it at? In the nucleus. DNA is found in the nucleus. And how is it different from RNA other than uracil? But say that, because it's double-stranded, right? So what is RNA? Single-stranded, that's right. Okay, so now inside this nucleus, on the sideline, okay, we're going to start to give the direction. Didn't quite make this big enough. Okay. Okay, now we're going to encode a message on a single strand called messenger RNA. Okay? So what is that? RNA polymerase is looking for a very specific spot, and it's going to start reading this process. And that process, when it is read, is called what? You were not translation. Transcription, because remember, scribes, we said, can read. So on our messenger RNA strand, what is going to go complementary to these pairings? No.
U.
Oh yeah,
U.
U,
I'm at A.
C, G, A.
Okay. So here is our play, if you want to call it that Coach Johnson has the instructions right here. Here's Wyatt, that he is reading what Coach Johnson told him to do. Now, this eventually stops. Where does our messenger RNA or Wyatt go now? He's leaving, and he's going towards the huddle, because on this strand, what does it have? The directions, right? He's got the plan. Okay? So then, what happens is, as we start putting these in here, 1, 2, 3, 4, 5, and 6. So we've got UAC, and then GAAC. G, A, and C. Now, what happens is, floating around in the cell. The reason we know that we're in the cell is because we're not in the nucleus anymore. We're outside of there. This whole thing is the cell, and this is one of the organelles that you find inside the cell. So, what happens then is, when you look at page 338, or maybe Mr. Wagner held this up, you can get the same information. Okay? So, this codon then is coding for which amino acid? You have to look at page 338 in order to tell me that. So, do we know how to read that table? Whether it's that one or this one, you get the exact same thing. So we look at here's our first one. So what you do is, on here it says your first letter. So here's U, okay? There's U. Then you go across the top. A is our second one. So we go across here, and A is what? Yellow color? Okay? Then along the right side, you just look for what your third nucleotide is, and it's C. So once you find U, then go over to the yellow for A, and then down to the C, you get tyrosine, UAC. Okay? So what that means is,
Just because you got yellow, how did you know to go to the C?
Because that's what it is. UAC.
it's right there. Okay? So then what happens, okay, we'll just look at it like this. Let's see, I think it's, what do you suppose that is, a 3-letter abbreviate? You said it. No. Tyrosine. So this is the amino acid tyrosine. So how does this know where to go to the ribosome? Well, it was on our messenger RNA. Remember, Wyatt has the directions. So therefore, what's coding for this is we have A, we have U, and we have G. So what you have here are the codons. That's got the code. This tells this amino acid that is what's going to be next. Okay? Then we got 3 more letters here. Remember, these 3 letters on messenger RNA are called what? It says it on page 338. What? Codons. Do you think you might know what these are called?
They are called
Anticodons. Okay? So what would our next amino acid be?
aspartate.
Okay? So we're going to put a C here, not T. It's going to be U. It's going to be G. So, again, we look for our blue. That's your first base. Okay? Then we look for U across the top. That is red. Then our third one was G, and that is I gotta look at the code again. C-U-G. So C, U, and G. It's called leucine. So then this is going to correspond up to here. So this has the code. This is telling these amino acids how they're supposed to be linked on here. This is a difficult process to understand. Okay.
So those deals are amino acids, and then like the adenine.
This
Adenine
just
above
means
that.
amino acid.
Yep, and then the adenine and the thymine are nucleotides.
Yeah, these are all nucleotides. It just depends how it is that you look at them. If they're 3-letter base pairings on messenger RNA, they're called codons. If they're 3-letter base pairings on transfer RNA, they're called anticodons.
How do you know if it's a transfer RNA?
It's because this is just floating freely around in the nucleus. Because if it was messenger RNA, it would have came from the nucleus.
So those are just 2 different things in that.
These here?
Messenger RNA yes, those are different things.
Yes. Now, this has a very monotone voice. Okay? I'm about to show you this video, and typically students like to see this about twice. It's only about maybe 45 seconds. So just brace yourself for this monotone voice. Okay?
Probably be screaming.
Probably what?
So probably be screaming.
No, it's not. And that's one of the things that, I don't know, myself as an educator, I try to change, per se, the tone that we're using and how we're enunciating things rather than, okay, now we're going to watch a video and it is about, maybe about a minute long, and then if there's anything left for time, we're going to go and answer some more questions. So what's that like for you when you hear that?
I just want to fall asleep.
Exactly. Okay.
You like this picture I drew of you?
Okay. Sure.
Pretty good.
Okay. So to get the full effect on here, okay, I'm going to put this up here so we can actually hear everything that takes place for those who may tune in at a later time, and hopefully I don't slip and fall. Okay. All right. That's what happens when we get moved. Okay. Brace yourself. This will be okay.
Each ribosome has a large and a small subunit, and each subunit contains RNA from the nucleus. The 2 subunits join after the protein synthesis begins. The first step is to receive instructions from the nucleus. Another molecule of RNA called messenger RNA carries these instructions in a 3-letter code. The messenger RNA attaches itself to the small subunit of the ribosome at the first site that is coded for an amino acid. The amino acids are the building blocks of proteins, and they are found throughout the cell. An amino acid is activated by the release of bond energy from a molecule of ATP. Now, a third type of RNA, transfer RNA, attaches to the amino acid. This molecule of transfer RNA contains the code for this specific amino acid, and it will carry it to the ribosome. The transfer RNA links up with a messenger RNA on the surface of the ribosome. The large and small subunits join, and a second amino acid is brought in. A bond forms between the amino acids. The transfer RNA moves off, and the whole arrangement moves down one space. Another amino acid is brought in, a bond is formed, and the process continues as the messenger RNA moves across the ribosome like a ribbon through a typewriter. The rate of protein synthesis is rapid. 10 new amino acids are coupled to the chain every second. Yet there is rarely a mistake.
Okay. Is it worth your time to see that again?
But I have a question.
Yes.
So, this has always happened, right? And it never stops or what?
This
So
is
there's just a huge long chain of our bodies, like, it just continues to get larger and larger.
There's actually a lot.
Okay.
From the moment when you're born, like,
Yep. It's a process that never stops.
So how big is one?
so
of
when
these
does it start? Like when you're in your mom's stomach, or when you're like?
I hope you're not. I know what you mean. I'm going by what you mean, not by what you say. Okay. I would venture to think that it probably does not start for you specifically as an individual until the heartbeat starts. Yes, you're going to have things that are starting to develop, but it's probably more of a tough question that I probably really shouldn't try to answer because I don't want to give you bad material. Okay. So, tomorrow, okay, because this is what we want to show you. Are we mature enough? Are we mature enough to handle this? I'm going to ask you again. Are we mature enough to handle this?
Yes.
How can you know what it is? Because I haven't showed you yet.
Well, that's why I was asking. Can we see it? Hey, why are you looking up there like that? Yeah, he's trying to-
Okay, we will continue with this at a later time. So hopefully I can get this placed on the Science Channel because this information is important for our curriculum. Okay.